This article is titled CRISPR/Cas9 for Cancer Research and Therapy. This article focused on the use of CRISPR/Cas9 against cancer and how it has opened up new avenues for cancer research. In the abstract, it says CRISPR/Cas9 and modified versions have found a widespread use to engineer genomes and to activate or repress the expression of genes. As such, CRISPR/Cas9 promises to accelerate cancer research by providing an efficient technology to dissect mechanisms of tumorigenesis, identify targets for drug development, and possibly arm cells for cell-based therapy. Cancer is said to be one of the main causes of disease-associated mortality, with the rising incidence worldwide. The progressive knowledge of tumor biology has led to the development of small molecules and antibodies targeting key proteins of oncogenic signaling pathways. This has helped in aiding certain types of cancers, but doesn't seem to work in all cases. This is where the idea of using CRISPR/Cas9 has come into play because of its versatility and how it can be used. CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats, is a family of DNA sequences found in the genomes of prokaryotic organisms. It was first found in E. coli in 1987 and then later on in other bacteria species. Microbes use CRISPR in order to protect themselves from invaders such as viruses. The microbes will capture snippets of the DNA of the intruder and store them away as CRISPRs. If the same invader/virus tries to attack again, those DNA segments that are turned into short pieces of RNA help the enzyme known as Cas find the DNA of the invader, which results in Cas splicing the DNA of the intruder. In 2013, the type 2 Cas protein from Streptococcus pyogenes, or SP-Cas9, was used for RNA-guided DNA cleavage in mammalian cells for the first time, laying the basis for using CRISPR/Cas9 as a widely applicable genome editing tool. sgRNAs, also known as single-guide RNAs, help function as a guide for targeting proteins which they form complexes with. So these sgRNAs help Cas9 and the modified versions of it get to the desired DNA. CRISPR has two RNAs, the CRISPR targeting RNA, cRNA, and the transactivating RNA. Both help to activate and guide Cas proteins to bind viral DNA sequence and are therefore particularly attractive to be used as a tool for genome editing. Certain modifications to the Cas9 protein has allowed it to cleave better to DNA in order to eventually cut the DNA. SP-Cas9 protein, which is an example of this because of how it has been modified to develop D-Cas9, which can be used to a variety of effector domains to mediate specific local DNA manipulation. CRISPR interference and activation exploit fused transcriptional regulators to repress or induce gene transcription when D-Cas9 is directed to the transcription start site of a target gene. CRISPR screening, which is a method used to enable thousands of genes to be modified and their functions assessed in a single experiment, is done by using pooled DNA oligos. A pool of oligos is designed to target a massive number of genes. Genes for drug resistance or for drug sensitivity can be identified. Negative screens determine genes conferring resistance, and positive screens determine genes conferring sensitivity. CRISPR is said to provide a tool to manipulate non-coding genes of the genome and will accelerate the functional exploration of a so far poorly characterized aspect of the cancer genome, which useful to the future of the cancer research and future improvements of viral and non-viral delivery methods will be necessary to improve the in vivo application of CRISPR/Cas9, laying the ground for the therapeutic use of CRISPR in the future. Although CRISPR/Cas9 looks to be the future of developing treatments and therapies for cancer, it has its limits, which are low editing efficiency in tumors and potential toxicity of the currently available delivery systems. However, even with these limits, CRISPR/Cas9 is looking to be a big factor in the improvement of the cancer research that we currently have.